Chapter 14 starts out phasing us into general or human genetics from general genetics. First thing we need to talk about is human genome. So a genome is all of the genetic information that an organism carries. So there's a fruit fly genome, there's a dog genome, there's a human genome, there's a maple tree genome. And that's the study of that genetic information starts with chromosomes. Those chunks of DNA that contain many genes. And these are, again, just as a review, found in the nuclei of eukaryotic cells. Molecular biologists photograph cells in mitosis when the chromosomes are condensed and easy to view. This is a karyotype, what you see here, and as you can see, in humans, there are 23 pairs of chromosomes. I don't know how well you can see it, but they're numbered pairs, 1, 2, 3, so on and so forth. They're organized from largest to smallest. 1 is the biggest chromosome. And actually 21 is the smallest chromosome, but almost from largest to smallest. And then in the bottom right, we have our sex chromosomes. X chromosome, Y chromosome. Chromosomes are organized and grouped based on size. And then what you see here are these colorful banding patterns. And as you can see, these are the homologous chromosomes we've been talking about. I just circled chromosome 16. The banding patterns are the same. The size of chromosomes are the same. Now, it doesn't mean anything. Like here with chromosome number 1, we have one chromosome that's kind of straight up and down here, and then the other one that's kind of bent like that. That means nothing at all. It's no big deal. It's just what we look at when we're looking at chromosomes to make sure we have the correct number of each type of chromosome, which is 2, and there aren't any extra or missing pieces, and we determine that by looking at the banding patterns, like you see here. This is kind of a review of what I just said. Karyotype is the diploid set of chromosomes grouped together in pairs, arranged from largest to smallest, 23 pairs in a human karyotype. In the lower right of the karyotype, we have the sex chromosomes. Females are XX, males are XY. We did a Punnett square here on this slide. With the sex chromosomes. So we have the female XX up here, male XY down here. And when we separate those chromosomes into gametes, this is why when you have a pregnancy, there's a 50-50 chance of that child being female and a 50-50 chance of that child being male. Interestingly enough, all human egg cells carry a single X chromosome. That's the only possibility that a female can give. Males can either carry an X chromosome or a Y chromosome in their sperm cells. So it's true that males will determine the gender of a baby. More than 1,200 genes are found on the X chromosome. We have Duchenne muscular dystrophy, a genetic condition, the X inactivation center, we'll talk about that later. Hemophilia A is a blood clotting disorder, and color blindness. The human Y chromosome is much smaller, and it contains about 1/10 of the amount of genes, 140 genes. And most of them are associated with male sex determination and sperm development. So those two are the sex chromosomes, 2 out of the 46. The rest of them are called autosomes. Our shorthand to represent females, 46XX for a normal human female, 46XY for a human male. When we look at human genetic disorders, a lot of human traits follow a pattern of simple dominance. MC1R, homozygous recessive alleles, would give a person red hair. A person with red hair, like I said, has 2 of those recessive alleles. Dominant alleles help produce darker hair colors. The Rh blood group is another simple dominant or recessive. When you have a blood type that is distinguished as positive or negative, it's the Rh factor. It's just abbreviated, positive or negative. Positive is dominant, so an individual with both alleles will have positive blood. Rh negative blood is found in individuals with 2 recessive alleles. That's a typo there. It would look like this. Rh negative, like that. Codominance is found in blood type. The ABO blood group has codominant inheritance, and also, this is an example of multiple alleles. So there are 3 alleles. There's the IA, IB, and I. I stands for immunocompatibility. Is it compatible with a person's immune system? So this chart sums up blood grouping and the things that, the concerns that have to be taken into account. So on the far left here is the phenotype. Person with blood type A can either be homozygous for the IA gene or heterozygous, IA and then little I. This person can donate blood to a person with type A blood or to a person with type AB blood. Can receive blood from type A or type O. Here's the example of codominance. Both forms of this gene are expressed. The identification tag on the red blood cell is both A and B, whereas in blood type A, just the A markers on there, B, only the B markers on there. Now, with blood type O, which is recessive, there's no identification tag on the outside of the red blood cell, so type O blood cells basically fly under the radar in an immune system. So type O blood can donate to all the other blood types, but can only receive blood from its own. So it's often said that people who have type O blood, or that blood type O is the universal donor. And people with type A blood, type AB blood are the universal acceptors. So if a patient has AB negative blood, it means that the individual has IA and IB alleles, and then 2 Rh negative alleles. I just kind of explained this. These antigens are what I was referring to as ID tags. Individuals with IA and IB both produce A and B antigens, making them blood type AB. And then, like I said, the lowercase I allele for type O blood is recessive. We went through this already. Blood type A, 2 different genotypes can produce a phenotype of blood type B. Only one genotype can produce blood type O. It's another form of inheritance we're going to look at. It's called sex-linked. It's a gene located on a sex chromosome. Genes on the Y chromosome that are called Y-linked are found only in males and are passed directly from father to son. Genes on the X chromosome are found in both sexes, but the fact that men just have one leads to some interesting things. Humans have 3 genes responsible for color vision. They're all on the X chromosome. In males, if any of those 3 genes isn't working, color blindness arises. Red-green color blindness occurs in about 1 in 12 males. It's the most common form. In females, though, color blindness is only about 1 in 200. So in order for a recessive allele to be expressed in females, it has to be present in 2 copies, 1 on each X chromosome. It only has to be present in 1 copy in males because males only have 1 sex chromosome. So the recessive phenotype or the affected phenotype of a sex-linked genetic disorder tends to be more common among males than females. So if just 1 X chromosome will do the trick in males, then what happens to all that extra genetic information in female cells? In the case of females, most of the genes in one of the X chromosomes is randomly switched off. It's called a Barr body, the dense region in the nucleus that's this inactivated X chromosome. And these aren't found in males because males only have 1 X chromosome and it needs to be active and working in order for them to survive. X chromosome inactivation happens in other mammals. In cats, a gene that controls the color of spots is located on the X chromosome. One X chromosome can have allele for orange, the other X chromosome can have an allele for black. So this is random X inactivation, and that leads to a cat's fur having a mixture of orange and black spots. Male cats that have only 1 X chromosome will only have 1 color of spots. If you see a cat and it has 3 colors in its fur, white with orange and black spots, it's almost 100% that cat is female. Last thing in section 14.1 that's talked about is a pedigree. Family history, relationships between members of a family. Pedigree shows presence or absence of a trait, follows it through parents, siblings, offspring. So in this case, a white forelock is like a person has dark hair and then a white spot at the front of their hair. All these squares represent males. All the circles represent females. These horizontal lines between males and females indicate that they've not necessarily marriage, but that they've had children together. So these top 2 people had 3 children, a male and then 2 females. Usually we put these in order of age, so it's oldest to youngest from left to right. And then we have these 2 people had 2 daughters. These people had 2 sons and a daughter. And then what we're following, this trait, you shade in the people who have that trait, and you leave open the symbols of the people who do not. So we see this trait passing from one generation to the next to the next. When you see a trait going from one generation to the next, we call it a vertical pattern of transmission. That indicates that that trait is dominant. So, this is the grandfather. 2 of his 3 children inherited the trait, and then 3 grandchildren also inherited the trait. The relationship between this woman and this male is aunt to nephew. This is father and daughter. Make sure you understand how to read the relationships in a pedigree. So because the white forelock trait is dominant, anybody who lacks the trait is homozygous recessive. One of the grandfather's children lacks the white forelock trait, so the grandfather must be heterozygous. So this is like a puzzle also. You're trying to figure out the genotypes of everybody involved. So we know that the people who are affected have at least one dominant gene. But we know that since this man has an unaffected daughter, he has to have a recessive gene to pass on to her. And then all the affected son and daughter also have to be heterozygous because of their mother's genotype. The information gained from pedigrees makes it possible to track how something's being inherited, dominant or recessive, autosomal or sex-linked, and determine the nature of genes and alleles associated with inherited or human traits.